but the composition of bacteria in your gut different from my gut. So how to make personalized medicine by making one yogurt that contain the same probiotic. So this is the bunch of different challenges we need to overcome to make that crazy dream come true. But I still wanted to dream high because that is why not? Because that may potentially help a lot of people become happier.
So hello and welcome to the Sheekey Science Show, where in this video I speak with Tae Seok Moon, Associate Professor of Energy, Environmental and Chemical Engineering at Washington University in St. Louis. He is an expert in synthetic biology and metabolic engineering and has published over 60 peer-reviewed papers, and also hosts the Synthetic Biology Young Speaker Series, himself also a YouTuber. Here we speak about his research and its amazing applications from degrading plastic to developing smart probiotics. I hope you enjoy our conversation. So, hi Tae Seok, thank you for joining me today and welcome to the Sheekey Science Show.
Hi, thank you so much for inviting me to this wonderful show. Thank
Thank
you.
you. And so, one of the reasons why I got interested in your research is that you're interested in synthetic biology. And so, I thought initially it would be good to ask what exactly is synthetic biology and what parts of it got you interested into the field.
So that is so challenging question because if you ask the same question to different people, you will get the different answers. I would say 1,000 people will have 1,000 different answers. So I've been in this field almost 20 years, but still I try to define the what is synthetic biology. So I honestly don't know. The field is still evolving in some sense, but if I am asked to define, I would say synthetic biology is some effort or fear that sort of engineer living system in a programmable way to achieve some practical goal, like solving global problem including climate crisis, medicine, and also food shortage and sustainable production, and also pollution problem, etc.
And so, how did you get interested in the field in the first place?
So that is interesting question. So if I go back to uh, let's say go back to the college year. So I major in applied chemistry, and I didn't take any biology course in high school except one basic course. And then somehow I fascinated by biochemistry. And then I started getting more coursework from another department that is a microbiology and biology department. And then I was so excited by the biology and also all the secret and mystery of biology. And then I interest in biochemical engineering because I'm the engineering department at the time. So I joined as a master student in biochemical engineering laboratory, and then I joined the company. And then I start to read a lot of papers, not necessarily my own, field in the company to broaden my horizon. And then two paper came to my life. That is actually seminar paper by Jim Collin that is about toggle switch published in Nature, and another paper published in the same year 2000 in Nature that is the by Michael Alloyd that is about the oscillating genetic circuit. That two paper is so simple, but also very elegant to describe living system, and that they call it genetic circuit, and then they implement that using very simple gene and put together, and then demonstrating some genetic, circuit like we see from electronic system. So that two paper basically fascinated me at the time, more than 20 years ago. And after I done my, industry experience, I wanted to become a PhD student. So I joined MIT, and at the time, there was no synthetic biology professor, but there was one professor working on metabolic engineering. That is one part of the synthetic biology, I believe. So I joined her, Kristalla Preder, and then I work on metabolic engineering, but still interested in hardcore synthetic biology, including genetic circuit building and so on. And then I lucky enough to be trained in the first center for synthetic biology in US, that is called Simberg. And then as a I call it Simberg kid, I met a lot of professor during the annual meetings. Uh, at the time, I taught many professors, including Jay Kiesling, Adam Arkin, George Church, all the big name in synthetic biology, and also Chris Voigt. He became my advisor for my postdoc work. And then in his lab, I learned a lot about synthetic biology, including genetic circuit building and refactoring. That is a process to simplify genetic system using similar concept we use in the computer science or electronics. So that's how I became synthetic biologist. And after my training, I start my position here 2012 in St. Louis, working as professor here.
I see. And so I'm a biochemist myself, not an engineer. So I'm kind of interested to understand what are the engineering principles you apply to build these circuits in the sort of biological form.
So there are two keywords in terms of synthetic biology. Why this is the different from previous biology disciplines. The first one is this one focus on quantitative understanding using mathematical model. So we use a lot of analysis, but mostly quantitative analysis, and then we use some mathematical model, and then analyze, and then after that analysis, we use that you know insight we gather, and then we build better model. In that way, that model predict the behavior of the biological system. So that's one keyword. And the second keyword I would say is application. So this is an engineering field that's a different from biology, because before biology is all about
Mm.
observation. See how bird fly, how animal gather together, but we just observe. And then if you wanted to observe inside, you probably remember you cut your frog, in high school or middle school, and then see what's inside. So this is all about the biology before. But now synthetic biology, we wanted to understand biology by building sort of living system or mimicking living system and analyze after building some small portion of the biological system. So that is the key difference. And then the goal is, at the end of the day, application to solve global problem, including, as I mentioned earlier, climate crisis, food shortage, and pollution, sustainable production, and also medicine. As you know, I mean, we already seen product of synthetic biology that is actually COVID vaccine, I believe.
Mm.
So that is two key difference between traditional biology and synthetic biology.
Yeah, that makes sense. And so, obviously, there's a lot of excitement about synthetic biology at the moment because, as you mentioned, for all of these different applications. And so, I know from your own research, you've done several different projects, but one of them's been identifying a bacterial strain that was able to break down plastic. And I was just wondering if you could talk
Mhm.
about that project and what that involved.
Sure. So our paper published, and that paper focused on PET. That is the, you probably already know. I mean, the PET bottle, you probably use a lot. But this is huge problem because this PET basically consists, I mean, this PET basically around 12% of all the solid waste in the world. I'm talking about 12% of entire solid waste. That solid waste include metal and plastic, some other waste from the food industry and also the pulping industry all together. And 12% is a lot because plastic is so light, but still weight wise the 12%. So we wanted to solve that problem, and the reason we chose PET because PET is probably the easiest plastic we could break down using simple chemistry. If you remember your chemistry, high school chemistry, PET is polyester, and the ester bond is easily broken if you add acid or base like NaOH, and then you just need to heat it up. And then if you heat it up to 150 degree Celsius, basically the polymer bond break by hydrolysis, and then that individual small molecule, we call it monomer, that is just chemical, but unfortunately one of the chemical has aromatic ring, and actually that is a little bit toxic to most of the
Mm.
bacteria. But our lab basically spent more than 10 years to identify some bacteria tolerate those kind of toxic chemical by using so-called adaptive evolution. That is basically the process you put bacteria into the some toxic environment or harsh environment, and then slowly increase the harshness or toxic level of the compound, and then improve the tolerance level of the bacteria. In that way, you could use that bacteria for example, bioremediation. It potentially clean up our environment. So that is what we done last more than 10 years. And then luckily, we chose the right compound 10 years ago. And then some of the bacteria among 2,000 different stock mutant in our refrigerator actually has ability to break down or utilize PT monomer. We If we break down the polymer into the smaller molecule, this bacteria use that monomer as sole carbon source, even though that chemical is toxic to other bacteria. So that's how we identify or discover new bacterium that utilize waste as a sole carbon source. And then this bacteria also amazingly produce some high value chemical, including lycopene. And I believe you already heard about lycopene. Lycopene is the one of the food ingredient coming from
Hmm. Yeah.
tomato. But we chose lycopene production by engineering or putting more gene or manipulating the gene in the bacteria to produce more lycopene, because I wanted to solve problem of plastic, but at the same time I wanted to solve the another biggest problem that is a food shortage or conceptually unacceptable practice that currently happening because many company produce lycopene by extracting lycopene from the tomato, but tomato should be something to eat rather than something to play with. As you know, probably your father or mom used to say, "Don't play with your food," but many companies actually play with food to making revenue. But at the same time, I want to say many people in developing country like Africa suffering from food shortage, and many people die at this moment. Not necessarily because they don't have tomato, but I believe conceptually using food to make some chemical that's not conceptually right. So we wanted to develop this technology, basically converting plastic waste into high value chemical like lycopene that might solve the two problems, including food shortage or food inequality, at the same time solving pollution problem that is because of the plastic waste. So that's one thing I want to mention for that specific paper you just mentioned.
Yeah, no, it's really incredible that it's not just consuming the plastic, but actually converting that to useful substances. In this case, lycopene, which, as you mentioned, is really high value. So now that you have this sort of like system established by using combinations of adaptive evolution and this somewhat genetic engineering approaches, how flexible is the system to then generate many other different compounds of high interest, and likewise using maybe different types of monomers to begin with?
Yeah, so that's a great question. So as I mentioned, PT alone basically comprise 12% of the solid waste. But still, PT is one of the plastic waste we need to deal with. So right now, since the first paper published, actually we started that project around 6 years ago. But several years ago, we transitioned after we see PT utilization or valorization or upcycling is working. We wanted to solve bigger problem that is a mixed plastic problem. I'm not sure about UK system, but in US we do not recycle well in terms of plastic. We put every recyclable plastic into one big waste bin. So separation is issue. You put polyethylene in that bin, polypropylene into the that one. There are too many packing material that's mostly polystyrene, and these are the key plastic waste, including the PET. So now we developed technology not only utilizing PET, but now we have ability to utilize PET plus PE, polyethylene, polypropylene, and polystyrene all
Wow.
together. And same concept, we break down that into smaller molecule. In that way, bacteria potentially eating those smaller molecule, but still those molecules toxic. But we have 2,000 different stock of the mutant bacteria or tough guy. The tough guy basically eating those plastic breakdown compound and then converting that into high value compound such as lycopene or potentially making biodegradable polymers including PHA, that's a biodegradable polymer, or we could even make some other polymer like nylon 66 or some other polymer using similar concept. The only thing we need to do is, of course, optimize the process in that way that's the economically viable. At the same time, we need to engineer the pathway, metabolic pathway gene. So rather than making lycopene, we also channel that waste, or that's the nutrient for the mycobacteria, into some chemical we or polymer we going to make. And to do so, we need to change some gene, like delete some unnecessary gene that kind of competing with my pathway, or adding some gene that has high activity. In that way, you making more product of interest. So that's our current status. After we saw those kind of technology, I actually start a company translate the technology into reality. So I'm so excited. So I form a company, and then the company name is Moonshot Bio. Why not? My last name is Moon. And then Moonshot is something we wanted to try, and then that's the Moonshot Bio.
Yeah, no, it's a good name, and it's really impressive the work you've achieved so far. And so, I mean, you make it sound somewhat trivial, but I imagine it's a lot of hard work. And so, I mean, what are the major bottlenecks that you face when trying to build these different systems?
So the bottleneck, of course, is there are many bottlenecks. So the simple bottleneck, or probably the most important one, is when it comes to making lycopene out of plastic. And current lycopene market is basically uh, food ingredient. That's a food supplement. We are so lazy or busy, so we rather than taking tomato, people taking some pear that contain a lot of vitamin and lycopene and so on. But the conceptually, this is very difficult to convince people to take the pear, but the ingredient coming from waste.
Mm,
So that's a market problem. But if you're thinking about another market, lycopene also used in the cosmetic company, because that's a red color, so cosmetic red color, and also antioxidant. So there are many good benefit having lycopene, but that is a one market challenge. But in terms of technological challenge, I wanted to say that is something promising, but this one is literally 6 years of effort to just show the demonstration or proof of concept demonstration at this moment. This one is work by all my student and postdoc, literally working every day and night, because they are passionate about solving global problem, including pollution problem. And technological problem is we demonstrate making small amount of lycopene out of the plastic, but the amount right now is tiny.
mm,
It's something like only milligram per liter scale. But to make that commercially viable, we should achieve minimum gram per liter scale in the final production level. So that means we need to improve minimum several hundred fold more improvement. But I believe, I'm also confident we could go there, because lycopene production studied more than 30 years. And then we already achieved more than 500-fold improvement by just using simple metabolic engineering technology. But with the technology advancement until now, from milligram per liter to gram per liter, that might be achievable, I would say, within next 30, 3 to 5 years time frame. But still, that require lots of effort, student hours, and postdoc spending day and night. And also, I also need to work with them to make that happen. But I'm very, very optimistic because all the young people in my lab working hard to solve this great important problem, and then that might be achievable in the future. And then potentially, if that happen, I retire early and then selling the technology at the same time solving all the global problem together.
Yeah, no, it'd be really cool to see that happen, especially within that time frame as well. But no, I realize this would be a lot of work. But obviously, you mentioned lycopene has like health benefits, and I know that. I mean, this is just one project of yours we've really discussed so far. You've also done a lot of research on developing like smart probiotics, and I was wondering if you
Mhm.
could elaborate on some of that research.
Yeah, so uh, that is my main project when I start my position. So my first grant proposal I have written is about probiotic, basically potentially curing parasite problem in developing countries such as Africa. So I submit that idea, and to Melinda and Bill Gates Foundation, more than I think almost 10 years ago, and then I got the money.
Nice.
And that was a super honor because Bill Gates even posted my project in his Facebook, and I even didn't know because I was not social media person until 3 years ago. And then I found out, because my friend said, "How do you know Bill Gates?" And then my answer was, "Everyone know Bill Gates. What are you talking about?" And then he said, "No, no, 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 no. I meant why he put your picture and your project in his Facebook." And then I was so surprised and honored at the time. And that is all about figure I made using Microsoft
I'm
PowerPoint.
glad it was Microsoft.
And yeah, of course. And then he probably loved it in some sense. And but the problem we wanted to solve is very important problem because many people, especially in Africa, especially young kid, infected by parasite, and then suffering from those disease because they don't have infrastructure in terms of the clean water and clean environment. So our idea is basically we wanted to use synthetic biology concept, probiotic potentially equipped with the sensor, the biosensor. That biosensor detect parasite, and then once parasite detected, probiotic generate some molecule that potentially kill parasite, by producing some chemical or some molecule that specifically killing parasite. So that was idea. And then since that we wanted to expand the same concept. You may be surprised if I say bacteria are smart, but bacteria are very smart because bacteria also sense environment. They don't have eyes or ears, but they or nose, but they still sense environment. Because of that, we could manipulate bacterial gene to make them to sense even human disease. And also bacteria also produce lots of molecule. For example, you probably already heard about antibiotics. Antibiotic actually produced by the bacteria. So for some disease, bacteria could also produce some molecule that potentially cure the human disease. So basically that's our concept. Bacteria once sense some human disease, and then bacteria produce some molecule for human. So to do so, I wanted to use some benign or safe bacteria, and one of them is a probiotic. So I want to ask, do you drink or take yogurt?
Yeah, on occasions I do, yes.
On occasion, okay. Yogurt is very good for the health without any manipulation, but my idea is I wanted to make yogurt that contain my engineered bacteria, because the typical probiotic in yogurt cannot sense disease, but I want to make this bacteria sense disease or some other symptom you may have. And then I'm going to give one example that is something interesting to many audience, I believe. Let me ask one question. Have you heard about neurochemicals or neurotransmitters found in your gut?
um, yeah, so I should clarify to my audience that we have had this conversation before, but yes, serotonin is one I know that our gut microbiome produces.
Sure. So serotonin is one of the great example. Serotonin you may heard about, but serotonin in fact produced by your gut cell rather than your brain cell. More than 90% produced by your gut cell. So that's very interesting or shocking fact when I heard that, you know, report many years ago, because I thought I'm human and all the neurochemical that control my mood, happiness should be produced by my brain cell, not by gut. And then I realized gut feeling might be true because serotonin produced by my gut cell more than 90%. Another important fact I want to mention is bacteria, total number of bacteria we have in our body, the number is more than much higher than total number of human cell we have. So you believe you are human, but you are human plus more bacterial cell you carrying every day. And then as you can imagine, you have more bacterial cell, bacteria influence us. And one of the way bacteria influence us is bacteria also have ability to metabolize neurochemical that found in your gut and also synthesize neurochemical. So after I thinking about this all fact, and then I come up with some crazy idea. So one day, if I successfully implement the idea, I want you to take my yogurt. That yogurt contain my probiotic, and then this probiotic sense serotonin, because serotonin is one of the important neurochemical control your mood and happiness. And then let's say your serotonin level from 30 to 100, that's the good level for you to be happy. And then bacteria sense, and if the level is between that right range, your bacteria now start to produce pigment, let's say the green color. If you see your poop, I'm sorry, I'm talking about poop again. If you see your poop after you take my yogurt, and if that's a green color, today is the happy day because your serotonin level is just right. And then you just enjoy your happy day today. Tomorrow, unfortunately, serotonin level also fluctuate, and tomorrow your level is too high or too low, and then bacteria start to produce in response to that concentration now red color, and today is unhappy day, So stay at home, don't do anything stupid, and just until your serotonin level become right level. So this is a diagnostic application. Your bacteria tell you by showing color in your poop, basically you are happy or you're not happy. That's one application. But why we stay there? Because you could have better or accurate measurement of your serotonin level, not just gut serotonin level, but even blood serotonin level. That's more important because that is basically affecting your brain. Even though gut level influence your blood level because that circulate or equilibrate, you could go to hospital and take your blood, and then a couple of days later you will see your result, but that's too probably late because that is only telling you 2 days ago's serotonin level. So what I want to do is real time detection of serotonin using probiotic. But my real crazy idea is not just detecting serotonin, but I wanted to control serotonin like we control temperature in the room. So in the room, we have thermostat, and if the thermostat working fine, if the temperature is too high, the air conditioner start to cooling down. But if the room temperature too low, and the heater start and then making the temperature rise. Same thing for the serotonin control. If your bacteria sense and then if bacteria produce enzyme that break down serotonin in response to high concentration of serotonin, you can make serotonin level go down or below the 100. That's our threshold in terms of happiness level. If the serotonin level is too low, you could make bacteria respond to that and then produce enzyme that synthesize serotonin. In that way, serotonin level become higher than 30. That is our threshold. We kind of determine. So having the same idea, we call it dynamic control or temperature control, the same way as the temperature control in the room. If that is possible, we could make your serotonin level always stay in the some certain range. And then I already have my commercial punchline, and then that punchline sound like, "Do you want to be happy?
Yes. Yeah.
Take my word, you will be always happy." So that's my punchline. But as you can imagine, this is a crazy idea, and then that takes a lot of years of research and optimization because each individual different in terms of diet and complexity of the microbiome in your gut, and then your level of the serotonin fluctuate. So you need to make the sensor that quick enough to respond to serotonin level, and then that is also fluctuating. At the same time, you need to make the bacteria survive or behave well with other bacteria, but the composition of bacteria in your gut different from my gut. So how to make personalized medicine by making one yogurt that contain the same probiotic. So this is the bunch of different challenges we need to overcome to make that crazy dream come true. But I still wanted to dream high because that is why not? Because that may potentially help a lot of people become happier. So that's my sort of crazy idea I want to improve.
Yeah, no, I mean, for sure, it's a really compelling idea. And I think this is, again, what attracted me to synthetic biology. It's just like how creative you can be. And obviously, once you've got the system in place or some way of getting the bacteria into the body through the yogurt in this case, you can just change the sensor and change the enzymes within the system. So it's not serotonin, it's all sorts of different molecules that it could be sensing and responding to. But as you mentioned, it seems like there are many other challenges associated with building this in the first place. Some of that's also probably a lack of our own understanding of what's happening in the gut microbiome at the moment, the different species
Right.
present in different people. But another challenge is also getting the probiotics to persist in the gut for a period of time to be able to respond dynamically to the changes. But then also, while you want them to persist, the other concern is you don't want them to persist necessarily too long or to escape and to start, you've got genetically modified bacteria. There's a lot of like safety concerns potentially about this. And I know you've also done a lot of research on biocontainment, bioremediation, and developed these really awesome kill switches. And so I think it'd be awesome to hear about that.
Sure. Thank you. So that's the brilliant question. So as you mentioned, biocontainment is a very important issue because all I try to build or construct is so-called GMO.
Mm,
And then even my wife and my daughter hate GMO. And I believe American is more generous toward GMO, but many people in European country, including UK people, literally hate GMO. And I also had one experience almost 10 years ago. I had debate with a famous former news anchor. After 1.5 hour debate, his conclusion was, I am making GMO and I am bad guy because I'm making a GMO. So that was conclusion. So since that, I especially interest in biocontainment because we watch too many horror movie. In that movie, basically bacteria become monster, even dinosaur or whatever monster, destroy the Earth or planet, and then we need to prevent that one. So since that, we try to develop multiple different uh, kill switch, I call it. The kill switch concept is something like this. As I mentioned earlier, this probiotic, let's say, successfully cure disease in your gut. The disease could be, some other disease, not necessarily only serotonin level control, but let's say cure some pathogenic infection by generating some molecule, or that could be some metabolic disease, because of the some molecule accumulate beyond the safety level. In that way, sometimes people die if you have some genetic disease. And then that is our mission we given to bacteria. Let's say the mission accomplished. Unfortunately, that's still GMO. And then the GMO will be released into the environment with your poop. So in that scenario, or the other scenario would be bacteria also mutate, because that is what we see in the movies, and then that may happen in your gut
mm,
even before your mission accomplished. There are many beneficial bacteria in your gut, but how can we make the specific bacteria we generate or GMO kill in a targeted way? So that was the question I want to answer. So recently we demonstrated after 7 years of failure, because we fighting against mutation, because we try to kill bacteria always find some way to mutate their gene or the kill switch, in that way survive. But main idea is something like this: if bacteria become bad guy, we could give some chemical that specifically target our bacteria and then bacteria start to die. And that's the main concept. But as I mentioned, we constantly fail because mutation always happen, and then our bacteria somehow survive, and then bacteria kind of propagate, basically making more bacteria, and then kind of dominate the population. Let me ask the question to you. Let's say we start 10 to the 11 cell per ml, and then let's say I kill all the bacteria except one bacterium. To give some idea, 10 to the 11 cell is a lot of cell. If you doing my biology research, microbiology, you probably heard about OD 600 for E. coli. OD of 1 is 10 to the 9 cell per ml ish. But now I'm talking about 10 to the 11 cell. That's 100 times more than you typically see in the laboratory. That is only achievable by doing high glucose fermentation, very dense E. coli culture. If I talking about 10 to the 11 cell, going down to the one bacterium, is it success or failure?
Well, I mean, if there's one surviving, it can grow, I suppose, but
So one surviving is not success.
exactly.
Okay, let me give some another idea. The US NIH guideline is if you start with the 10 to the 8 cell, and then at the end of the day, you have only one survivor, they are okay with that. But I start with the 10 to the 11 cell, that's a 3 order of magnitude higher, that meet NIH guideline, but you still think that's not success.
Yeah, I guess so.
Right. So that is probably correct answer because scientifically, last 30 years of research, GMO, because GMO has additional burden, that is not their goal, because bacteria do not care about our goal, because bacteria do not care about helping us. Bacteria do not care about curing our disease. Bacteria only wanted to survive. But we put some curing molecule and sensor, that's an additional homework they want to get rid of by mutating, right? So that's why we struggle, we constantly battle against mutation, and then that's how we almost fail every single time last 7 years because of mutation. However, because your bacteria has additional burden, the survive, the ability to survive in the real environment typically almost zero compared to the native bacteria in the environment. That is 30 years of data. However, as you mentioned, public do not see it that way because we watch too many movies and we don't know what would be the consequence this one bacterium in the environment. So as a scientist and engineer, we need to responsible for what we're doing. So I wanted to completely eliminate even just one bacterium, even though we start with the 10 to the 11 cell or even higher number of cells. But as I said, that's a very difficult problem. So my student Austin, constantly fail because we done multiple different approach to improve killing efficiency, but always there are another mutation happening in the kill switch. And then one point I was sitting in the hospital waiting for my doctor for 2 hours because somehow US hospital system is sort of messed up. So even though you book your spot, you need to wait 2 hours. So I have nothing to do except daydreaming. And what I realized at the time, or after crazy daydreaming, I start to think about what happening if we lose electricity in this hospital. The patient who almost died under operation, doctor could do operation. The answer is fortunately yes. In most developed country, they have backup generator always kick in whenever we lose power. After thinking about that one, why not? We could have multiple kill switch, not only one but several of kill switch because mutation happening random. If somehow mutation randomly destroy kill switch, we still have several more kill switch to act. So after we putting total 4 kill switch, even though 3 was sufficient, but I want to have one more. In that way, we make sure extra one more probably doesn't hurt. But technologically, that was very challenging, but we achieved it. And then after we have 4 kill switch, and we tested that kill switch in the mouse model, now all the bacteria we provide into the mouse, and then mouse, if we give the specific chemical, the kill switch containing bacteria all die regardless of the number of cells we originally putting in. At the same time, we wanted to make sure bacteria do not survive in the environment. To implement that idea, we wanted to use the difference between our temperature, that is typically body temperature is typically 37, and outside that typically a little bit below or around 30 degree even during the hot summer up in Africa, because at night temperature go down. So the idea is basically bacteria come out of your body and temperature drop, and then your temperature sensors basically detect the temperature downshift, and then basically bacteria start to produce kill switch molecule. In that way, bacteria just start to die. So we demonstrated using, again, my favorite subject, mouse poop, and we collected, and then we demonstrate in the 30 degree and the other into the 37, and then only we saw survivor at 37. That is kind of mimicking the same temperature in the body, but we didn't see any bacteria at the 30 degree because the kill switch basically kill all the bacteria.
Nice. So you mentioned you had up to 4 kill switches, and so are these orthogonal? So you have chemicals as one of them, temperature as another, and so what are the different sort of signals that can trigger the kill switch?
That is a great question. So we use the either chemical or temperature. Also, we use both of them together. At the same time, this kill switch is orthogonal because their sequence is different. That means even if your mutation process somehow destroy one kill switch, you still have the 3 more other intact until you destroy all 4 of them. Your kill switch is basically working. I didn't explain all the detail how the kill switch working because that might be too technical. But to give some idea, we use so-called CRISPR system. You probably heard about the name CRISPR because that is the one topic, Nobel Prize, basically given. But CRISPR is the molecular scissor that cut DNA. Human and eukaryotic system all has so-called non-homologous end joining mechanism. In that way, even if your DNA cut, DNA join together and then repair themselves. But unfortunately for us, most of the bacteria do not have so-called non-homologous end joining mechanism. That means if you cut the DNA, the genome of DNA, bacteria cannot stitch that together, and bacteria, in theory, die unless the kill switch or CRISPR system has mutation that make the malfunction of kill switch. So that's the basic idea. So as long as we have intact one kill switch exists, always the cutting happen, and then bacteria die.
Nice. Yeah, no, I see. That's really cool. So what, like, I'm very interested in like where your sort of creative ideas come from, and sort of like how do you go about reading literature or like synthesizing ideas from different areas together?
So how I come up with the crazy idea? That's your
Yes.
question. Sure. Uh, because I'm the crazy guy. That's the short answer. No, no, the let me put it this way. So I used to be very shy, you know, thinking about things very in a safe way. So I was typical average professor, before. But one event completely changed my perspective. So what happened was, I was a professor, and then one of my student actually coming from Africa, but unfortunately, he was black guy, because of his race, he was sort of discriminated by the leadership people in the university, and he's almost kicked out of the university, also almost kicked out of the US. And at the time I was untenured professor, I decide to keep quiet to protect myself, at the same time protect my student. That was just my excuse, I would say. And since that, I was so ashamed of being professor because I could not speak up at the time to save my family or my career. And then I became so sick for many years, literally dying for almost 4 years. And then in the year of 2020, when everyone suffered from COVID, I didn't care about COVID much because I thought that in that year, I thought I just die anyway, regardless of COVID, because of my symptom and pains. That is combined pain from mental stress, at the same time real physical pain every single day or every single second, almost 4 years. When you face your death, I guess you could do whatever you want to do, including something you never imagined. So that year, before I die, I wanted to put my crazy idea. I thinking about, let's say, more than 10 years, but I never put into the reality. So I put five proposal, collecting all the crazy idea together, and then submit it, hoping that is selected after I die. And interestingly, I miraculously survived in some because of some event, and then all five idea was funded.
Nice.
And then since that, I realized I want to solve many global problem. I believe total 10 problem I want to solve, but just incremental advance of knowledge cannot solve this biggest problem we currently facing, including climate crisis, foods shortage, sustainable production, and also medicine problem or pandemic. I believe after that we should have some innovative or potentially disruptive technology should be developed to solve many of these kind of problems. So since that, I only thinking about crazy problem or crazy ideas because the small project can be done by some other people, but somebody should be crazy to show some potential disruptive solution. So that's how I you know became a crazy guy in some sense.
Well, I think your ideas have a lot of backing to them, and they're very exciting. So yeah, no, I'm glad that you've pulled through and got funding for these different projects. I'm curious as well, though, to hear, uh, like, what sort of advice would you give to other students who are interested in joining synthetic biology as a field, or where should they go to find out, uh, like, to come up with ideas, or what should they be studying?
Sure. You know, I have though just one advice. As I mentioned earlier, I've experienced very dramatic, life experience that start from discrimination and harassment based on race. But I'm not the only one. I believe many students and international scholars or just young people in general, they constantly worrying about things because the world we currently living in, there are too many sad news or gloomy news, including wars in including the Ukraine, Israel, Palestine, and also hunger in country like Africa countries. And there are too many other things. Also, simply you just broke up with your girlfriend or boyfriend, and life is full of crazy, sad things happening to us. But I want to say you are not the only one having those kind of things. But at the same time, you also remember happy moment in your life because of something you care about. I'll give one example that saved my life in some sense or partially, and that's great story. And then to give my advice, the student I just mentioned, who originated from Africa, discriminated because he is black guy. So basically, racial discrimination and harassment. He suffered from that one, and then have difficult time for many years. Almost kicked out of the school and almost kicked out of the university and country because he didn't have any power. But sort of I proud of myself because I protect him in some sense, sort of jeopardizing my career as well. And after he endured that all the difficulty, he finally managed to join a company. I believe you already know the company that is the Pfizer. And then he joined the special task force by utilizing all the knowledge he learned from the university from me to develop the first COVID vaccine at Pfizer, and then he became a world hero,
Wow.
or at least hero known to all the African continent people who interest in research in biotechnology. Because of him, I'm getting too many emails from Africa. for your information. But one thing I want to tell you is one great thing I learned from him was he has much more difficulty than me. For example, his home in Africa when he was studying here didn't have electricity, let alone internet. His mom suffering from cancer, still suffering from cancer. But he saved his salary even to send the money to his family. In the US, graduate student salary is very small, but still he saved the salary to support his family. And he endured all the difficulty you may face. But at the end of the day, what he done differently from other normal average student, that was he dream high and he never give up. And then he basically had one opportunity to join company and then to join the special force team. They work every day and night, and then they deliver the first COVID vaccine to the entire world, contributing to the society, probably the one of the biggest problem we face in recent years. So my advice to you is you may have lots of different problem, difficulties, challenges, or injustice, whatever you're facing right now, or simply you just broke up with your girlfriend or boyfriend recently. But that is not the end of your life. Just try to learn something from your experience or even failure, because failure would be the best teacher for your success in the future. And then dream high, because I saw many people, especially young people, suffering from those kind of difficulties, but still they living, hoping better future. I know because I traveled too much. Last year I traveled more than 160 days just to give seminars and meeting people. I did the same thing this year, more than 150 days. As you can imagine, I met a lot of young people. And even though I mentioned all the gloomy, sad news earlier, but I was so optimistic. I am so optimistic at this moment because of you, because I saw the future and potential and passion to solve all the global problem that could be injustice, hunger, or climate problem, or whatever problem you care about. Because I saw huge passion from you. You want to solve this problem, and then if you solve that problem, we'll have the better world 20 years later. Potentially, we don't have those kind of problem, or at least we solve many of the problem. But that all depend on you, and then you should be proud of yourself because you're still living despite of all the difficulty, and then you will become a better leaders to make this world better place. In that way, I will benefit because my daughter will live in a better world because of you.
Yeah, that's well
Uh,
said.
One sentence: dream high, never give up, and failure will teach you and make you stronger.
So true. It's just been incredible to talk to you, Tae Seok. You've been an incredible role model, and you're doing some very worthwhile research yourself. And I can only wish you the best of luck with all of your endeavors. And hopefully, we'll see the moonshot at some point. So yeah, it's just been great talking to you, and I wish you the best of success.
Thank you. It's my great honor to talk with you, and I hope to see many global leaders in the future among this audience.